Okay, so to understand the atoms and the moleculars, I think periodic table is very important. I believe all of you should already know the table. And then all of the chemicals, because of they have a different kind of chemical bindings, they have at least four major type of binding between or among different moleculars. And then as you know, or you can see in this PowerPoint slide, for the covalent bond, which is the most strong binding between two different atoms. So, and then the electrostatic bind is only 30 counts per molecular. So it's kind of compared to the covalent bond, is the weak covalent bond. And then as you know, the hydrogen bond, which is very important in our DNA structure, because all of our nucleotide like AT or GC pair was made by the hydrogen bond. Therefore, it's also a very important chemical binding, especially between the double strand of our DNA structure. And then you can see their energy actually is quite low. It's only 5 car per molecular. So it's the reason why we can easily manipulate, we can easily handle our DNA. We can easily, by using the chemical way or just give the energy like heating with our DNA, then we can denature the DNA from double strand DNA became single strand DNA, right? So in our lab, if we need the single strand DNA, especially like when we're talking about the hybridization reaction, basically we need prepare, especially for cDNA microarray, because cDNA is double strand. Okay, cDNA microarray, which is originally is double strand DNA. So we need to denature all of our double strand DNA become single strand DNA. Therefore, we need to put this kind of DNA into like boiling water, which over 90 degree, the water over the temperature of the water is higher than 90 degree. Therefore, the DNA can be denatured, became single strand DNA. All right. Van der Waals bonds, which is even weaker chemical binding among different moleculars. Okay. So, and then the electrostatic bond, which is also important when we prepare our microarray slides. As we know, the DNA carry negative binding, negative charge. So when we try to put DNA onto the microarray chip, so we can use electrostatic bond to allow DNA temporarily bound on the surface of microarray chip. Therefore, in order to do that, we need to coating some material, particular like polylysine. So you can check the charge of polylysine, which they carry a positive charge. So after we coating polylysine on the surface of the microarray slide, which is a glass slide, therefore the surface of the slide actually became the positive charge. So for our DNA, actually when we put our DNA onto the surface, then the negative charge DNA can be bound by this electrostatic bond onto the surface. Okay, and then you can see that the energy between the electrostatic bond is only 30 car per molecular. It's not a very strong bond. Therefore, even with the DNA can be bound on the surface, still can be removed. Like when we're doing the hybridization or when we're doing the wash reaction, then the DNA could be removed from the surface. Therefore, we need to provide more energy allow DNA to become covalent bond onto the surface of the glass slide, onto the surface of a molecular chip. Right. So you can see if it can become the covalent bond, it has a highest, strongest energy between the covalent bond. Right. So it's very, very difficult to be removed. And then like here, you can see the charge between negative charge and then positive charge. It can form the electrostatic bond between these two molecules. So that is very, very important when we put, especially for us to prepare the microarray slide. We put DNA, which is negative charge. It can be bound onto the polylysine because the lysine, the end of the functional group of the lysine, which carry the positive charge, so it can be bonded together. Okay, and then you can see here, which is a hydrogen bond. Okay, and then it's very, very important because the hydrogen bond only can be happen between the atom H and then the oxygen. Okay, so most famous hydrogen bond which is can be observed between the within the DNA. So hydrogen bond, which is the very important bonding, allow our DNA from single strand to become double strand. Okay, so here is the van der Waals bond. It's very, very weak, easily to be break. And then the surface tension, which is a high quantity microarrays require non-uniform spot diameters. Like if you Google microarray, okay, you can find a lot of microarray image from Google, and then most of them actually is quite beautiful because most of the microarray image they provide a uniform spot. Right. Therefore, to form a uniform spot, the surface tension is very, very important. That is the parameters that can be controlled by the surface tension, right? So so you can see here, because most of case our DNA have to be solved in the printing buffer, and then like in our lab we use water-based printing buffer to print our microarray material onto the glass slide. So when we prepare our glass slide, we not only to provide the charge for electrostatic binding between the surface and then the DNA, but also that kind of material can form the hydrophobic characters, allow to build up some certain of surface tension that allow our water molecular can perform a uniform tension, unlike this kind of without any hydrophobic character. Therefore, it's no especially hydrophilic, like this kind of hydrophilic and then this kind of hydrophobic character. So the water can be formed, okay, and then the stronger hydrophobic, then the more beautiful uniform spot can be formed. So in microarray, actually capillary reaction is very, very important, but that is the method that we can carry our printing targets. Then we transfer all of our printing target by printing pins onto the microarray surface. So over here you can see this is a printing pin. Actually, it's very kind of stainless material, and then on the top of the printing pin, if we zoom in the tip of the printing pin, actually there is a very, very thin channel, and then if we put into the prepared cDNA or whatever water, then the water can be held within this channel. Then we can use gravity to print, to transfer all of the molecular onto the printing area, which is the surface of the microarray chip. And then which is also very, very important, because, for example, we need to print more than hundreds, more than thousands, even 10,000 spots. So during the printing, we need to make sure each printing spot, they have the very unique and uniform the shape and then volume. Okay, so how to deliver the molecular the cDNA equally onto each spot is also very important. So that's the reason why each printing print is so expensive, because they have some special coating to hold all of our printing target that allow printing print equally deliver printing volume onto each spot. Alright, and then we also need some particular functional group, right? So we can see here we have alcohol, aldehyde, carboxylic acid, triol. There are lots of different functional group. So over here I'd like to introduce two terms like the nucleophiles, electrophiles. So the nucleophile is a molecular functional group, or the atoms that contain free pairs of electrons or a strong negative charged ions. And then for the electrophiles, it's a molecular functional group. It's a molecular functional groups, all the atoms that bear the positive charge, strong positive charge bonds, and then this electron sinking central bonds with neutrophils. So because of the chemical reactions allow us to make two different molecules that bind together. So that's how we coded. We can use this kind of features to coded some particular molecules on the microarray service. Like we can coding like aldehyde functional group onto the service. Okay, and then because of the aldehyde functional group on the service allow us the DNA, and then the DNA also coded the particular functional group, and then became the neutrophil to bind onto to form the covalent bond onto the aldehyde coupling molecules. Okay, so over here, we coded the aldehyde, we coded material, and then the end of this material carry the aldehyde functional group. And then when we prepare our DNA, and then the end of the DNA also coded the functional group. Allow these two functional groups can form the new binding. Okay, so actually it's quite similar like the epoxy end. So that is the very common microarray service, printed microarray service. We can coding aldehyde to allow the DNA to form the covalent bond, or we can also we can form the aldehyde, or we can therefore the DNA carry the functional group can form the covalent bond. Okay, or we can use the amine functional group. That amine functional group actually carry positive charge to form the charged service, allow the negative charge the DNA binding on it by the electrostatic bond. Okay, and then the other chemical will be used in the microarrays, the labeling dye, because when we doing the microarray experiment, actually we need to measure their binding level, okay? And then if you have more DNA binding on the specific target, it means you have more genes expression level. So but unfortunately, actually DNA cannot be observed. Therefore, we need something allow us to observe DNA. So, like when I was a student, we use radioisotope material to label DNA because radioisotope, they have a signal actually can be observed by the X-ray film, okay? And then in microarray, we can either use radioisotope material for the labeling, or we can use labeling dyes, okay? We can use tracing or Sci3 or Sci5 to label onto the DNA, and then the labeled DNA can be observed by the scanner, okay? And then because they have a specific emission wavelength, it's like the Sci3, the emission wavelength is 550, Sci5, which is the 649 nanometers wavelength. Okay, so it's quite, we can use, the emission wavelength actually is quite different. Therefore, we can use Sci3 to label one sample, Sci5 to label the other sample. Therefore, we can distinguish two different samples on one microarray chip. Okay, so that's a very, very convenient tools allow us to label two different DNA samples or two RNA samples, and then we can observe two samples at the same time on the same location because they have a different wavelength. Like Sci3 actually is green color, Sci5 for Sci5, which is the red color. Okay, so as we know, green or red actually is quite different, so we can observe these two samples, okay, together on the same microchip. So, nucleotide is the main molecule within our cells, and then then there are four. So the structure of a nucleotide actually they contain a base, and then a base, sugar, phosphate group. Okay, so there are four different bases for DNA. There are adenine, guanine, cytosine, thymine. Okay, so this a nucleotide A, G, C, T for the DNA. For RNA, there is the adenine, guanine, cytosine, and then uracil. Okay, so that is four different nucleotides for RNA. A, U, C, G. For DNA is A, T, C, G. Okay, and then for the sugar molecular within the DNA, which is the deoxyribose. All right. So the ribose means there are 5 carbon molecular within the sugar molecular. Okay. So we named this is the position 1, position 2, position 3, position 4, and then position 5. So there are 5 carbons within these sugar moleculars. Okay. And then because of at the position 2, there is only H without oxygen molecular with here. Unlike the RNA, which is a ribose. You can see over here, especially at the position number 2, in RNA, in ribose, there is OH. However, in DNA, there is H. So that's the reason why we call deoxyribose, which is DNA. And then for RNA is ribose because there are OH in the position number 2. Okay. And then there is the other very important is position number 3. No matter it is DNA or RNA, is OH group at the position number 3. And then there is a very important features of the deoxynucleotide deoxyribose DNA, because of the H at the position number 2 allow DNA more stable than the RNA. You can see over here there is OH, which is a very active the functional group over here. So that's the other. This is the major one of the major reasons that the DNA is easily became fragmented because the active the functional group at position number 2, the OH group. Okay, so the DNA more stable than RNA because only H hydrogen at the position number 2. Okay. Okay, and then here you can see the structure of a DNA. So here is the structure of an RNA, and then here is the structure of the DNA, and then you can see here's the deoxyribose, the structure, the sugar structure, and then here is the base. So the base is located at position number 1. And then you can also see the phosphate group. There are 3 phosphate groups, like here, within this position number 5. Okay. So if there is a base, cytosine over here, and then there are 3 phosphate groups at position number 5. So here is how we call cytosine triphosphate DNA. So CT5. So that is cytosine. Okay, and then the triphosphate group. So it's called CTP triphosphate group. So this is CTP. And then, however, if there is only 2 phosphate groups, will be C diphosphate group. So it's CDP. And then if there is only 1 phosphate group, will become C monophosphate group. So will be CMP. Okay, so that's how we call our nucleotide. Okay. So here is a very important the structure of our DNA. We already introduced you the structure of a nucleotide, and then the nucleotide can be formed between 3' OH group and then 5' phosphate group. Okay, so they can actually to become a covalent bond between 2 nucleotides. So the nucleotide can use the phosphate group to bind onto the 3' OH group to form the covalent bond between 2 molecules. Okay, so here is the 3' end, and then here is the 5' end. So the 5' phosphate group to form the covalent bond at the 3' end. So that's the reason how we always say, okay, we have a direction of the DNA which is from the 5' to 3', because when we try to elongate DNA, there is always the added DNA, the added nucleotide have to be added into the 3' end. Okay, so over here the structure of DNA. Okay, so always 5' phosphate group to form the covalent bond onto the 3' OH group, and then after we have the single-stranded DNA, then the DNA can form the hydrogen bond between G, C, or the A, T. And then because of the structure of the base, guanine or the cytosine, you can see over here there is the H and then hydrogen or the oxygen. So the hydrogen bond only can form between hydrogen and then the oxygen. okay, molecular hydrogen and then oxygen, okay, and then hydrogen and then the nitrogen, okay. So here, because between nucleotide G, base G, I should say nucleotide G and then nucleotide C, there is 3 position, okay. So between these 2 nucleotides, they can form 3 hydrogen bonds. However, between nucleotide A and the nucleotide T, they can only 2 positions to form nucleotide. Okay, so there are only 2 hydrogen bonds. So that's the reason when we always say, okay, so the GC content is so important when we try to understand one DNA or one gene. It's because if the gene contains more GC, it means the DNA within this particular region, actually they have more hydrogen bonds. More hydrogen bonds, it means when we try to denature DNA from double-strand DNA to become single-strand DNA, we need more energy. All right, it's the same. When we try to anneal DNA onto some particular region, if the region have high GC content, it means we need provide more energy, allow these 2 DNA to bind together. Okay, so that's the hybridization, especially during the hybridization reaction, we need to provide energy, allow 2 DNA, 2 single-strand DNA, they can bind it to each other. Therefore, if the DNA contains more GC contents, it means we need to provide more DNA, we need to provide more energy, allow 2 DNA to bind it together. If they have low GC content, it means have a high AT ratio within this region. Okay, therefore it may not need as much as the energy compared to the high GC content moleculars. Okay, so especially when we prepare our probes or PCR primers, the GC content is a very, very important features. Okay, so we always like, if we try to design PCR primers, the GC content is roughly, when we design it, we will choose the content is roughly between 48 to 55% of the GC content, lower than 55%. We don't like high GC contents, because if there are the high GC content region, means we need higher energy, higher temperature, okay, to either hybridize together or either to denature each other. Okay, so the hydrogen bond between GC and an AT is very, very important. That's a very important features about our DNA structures. And then, so here is a structure of our DNA. We can see there are 2 strands. Okay, one direction is 5' from the other one. Okay, to 3', and then the other strand, actually the direction is from 5' to 3', and then over there, their nucleotide, their bases can form the hydrogen bond with each other. Okay, so if the AT pair, there is a GC pair, AT pair. So the sequence, we call it sequence complementary to each other. It means if there is an AT pair, GC pair together, it's a complementary to each other. If not, it's a mismatched. If yes, it's a matched. Okay, so it's a base pair. And then the diameter of this DNA structure is 20 angstrom. So I already mentioned earlier, like if we have the RNA sample and then we extract total RNA from cell or from tissues, okay, we'll have the RNA, and then most of the RNA actually are ribosomal RNA. I'm sorry about the typo. So the ribosomal RNA should be roughly around 75 to 85% of the total RNA, and then the transfer RNA would be the 10 to 15% of the total RNA. And then the messenger RNA actually is the most important RNA, I would say, because all of the gene will be transcribed become messenger RNA, and then only messenger RNA can be translated become protein. None from the ribosomal RNA or transfer RNA can be translated become protein. Only messenger RNA can be translated become protein. However, messenger RNA are only 1 to 5% of the total RNA, so it means actually we have very, very little of messenger RNA among the total RNA. And then it should be because when the gene are needed and then can be translated, can be transcribed and then translated become protein. Therefore, messenger RNA is the intermediate product, so it have to be transcribed and then will be disappeared once the protein has been generated. Okay, so that's the other reason why only 1 to 5% of the total RNA. Messenger RNA can only 1 to 5% of the total RNA. Okay, so here instability of the RNA and I mentioned earlier because of the structure, especially at the position number 2, there's an OH group instead of the H like the DNA. So it's OH, so therefore it's neutrophilic, and then it can be easily attacked by high pH and OH or the enzyme. Therefore, it can become a covalent bond over here. Therefore, it will break the RNA chain. Okay, so that's the reason why, unlike DNA, the RNA is more unstable than DNA.
So this is the arraying process. So we have to prepare DNA, right? And then we have to prepare our structure. Okay, in this case, it's a microscopic slice. It's kind of slide you guys been using it when maybe when you were in the not the elementary school, in your middle school, to see some microorganism under microscope. Okay, we're using the same thing. So these are the typical microscopic slides. It's 1 inch by 3 inch. Okay, 2.5公分 by like 7.5. So it's almost identical. Of course, we are using much better quality slides. Okay, it's quite expensive actually, not just the regular glass. Okay, so you have your DNA prepared, and then of course we're going to discuss that even further. You know what kind of productions, manufacturing procedure we will be using. So you prepare your DNA, ready? You have your slide. Of course, I probably mentioned that in my introduction lectures, saying that pure glass DNA won't bind onto it. You need to do something with the surface. And then we will discuss that later on, say what kind of surface chemistry you can use for doing this. So that's why I put a coating here, okay? Can be different kinds of chemicals. Once you have this ready, you have your DNA ready, and then of course this is our arrayers. As I probably mentioned that briefly. So array, okay? In Chinese we call it 打点机. Right? This is a 3-dimensional robotic system. So you can move either forward, backward, to the right, to the left, and then up and down. So you will be able to deposit whatever DNA you prepare here in the specific areas. Okay? So you constantly printing different DNA sample onto this coated slide using this particular robot. It looks like quite simple, right? Just XYZ, 3-dimensional robotics. But it's not that easy, especially the motion control. Okay, each individual slide you saw from here, it's roughly about 100 micrometer diameters. Okay, in Chinese, 它的直径只有100个 micron, which is a very tiny spot. That's why we call microarray. Right? Micro is very small, relatively small. So how can you print something on a specific area without kind of, printing in the wrong, maybe the spacing is very uniform, okay? So that requires very precision control of your robot. So you print the first spot, then you move to the second spot. Each movement has to be tightly controlled by this system. So this particular machine will cost roughly about 100,000 US dollars. Today's exchange rate is about 280万台币. Even though it's just a small 3XYZ robotics, that still costs a lot because they have very nice motion control. We're going to discuss that later on. Okay, so you put your slice here, use this 3-dimensional robotics, and then you print your DNA onto a specific spot on each individual slice. Okay, in this case, this one you can print 100 slides. So you can print 100 slides at a time. So once you finish your printing, usually we print 20,000 spots. Okay, 2万点. For 100 slides, it takes about 16 hours. 16个小时 to finish this a set. Okay, so early days, some of my graduate students, they are very sort of hardworking because they have to take different shifts to take care of these machines. At that time, we're still printing our own array. Okay, but nowadays, we don't do that because we're going to discuss about the commercial platforms. Okay, commercial arrays are way better than our own in-house printing. So anyway, for 20,000 spots, you print 100 slides, it takes about 16 hours. Okay. Once you have finished that, then we have to go a process called post-processing. Why we want to do that? Because remember, we use different chemicals to do the surface coatings, okay, or surface treatment. You will find out what's the difference in surface treatment and what is the surface coating. Okay, so you have to go through this. We will discuss that even more detail later on. So you have to go through this, be ready for testing. Okay, so this you add different chemicals and using different temperatures. Try to watch or inactivate some of the area did not have the DNA onto it. Once you go through this, and then you have to dry the slides. Okay, and then this slide then ready for some experiments. So again, we label with 2 different colors. So you have 2 different samples. Maybe one is coming from cancer cell, one is coming from normal cell. And then you want to compare what's their gene expression profile between these 2 cells. Okay? So in Chinese, we always say this, maybe this is a 实验组, right? This is a control, so we say 对照组. I want to wonder what's the difference in terms of their gene expression between two different cell lines. One is normal cell, one is a cancer cell. So you label them with different fluorescent dye. In this case, we use a red fluorescent dye and a green fluorescent dye. Okay? And then, of course, gene expression. When we were talking about gene expressions, it's two levels. One, usually we it's transcriptional level. It means messenger RNA. Or the other one we call it translational level, which is protein. Okay, so when someone asks you what's the difference in terms of gene expression, you may ask them specifically whether that you're talking about the messenger RNA level as a transcriptional level or that's a protein level translation. So in this case, of course, we are studying messenger RNA. So you have messenger RNA, right? We go through this reverse transcription to become its complementary DNA, right? Stand for C, so cDNA. In the meantime, we add fluorescent dye. Okay? We use green and red. So that's why you see this two color fluorescence here. Once you have finished this, then you take this array to do the hybridization. Once you have hybridization, then you finish this. Usually it's overnight. We set it up in the afternoon, and then next morning you come in and you wash off. And then you can put this slide into this scanner over here. Okay? And then of course on your computer screen you will show something like this. So you will see some spots with the red, some spots with the green, and some with the yellow spots. So based on their fluorescent intensities, then you will know what's the gene expression level between cancer cell and normal cell. Okay? For example, if you label your cancer cell with red, and then your normal cell with green, if you see a spot that's very red, what does it imply in terms of gene expression? It implies that particular gene is highly expressed in cancer cell versus normal cell, right? Because it's very red. So that means that red fluorescence is very strong. Green intensity is much, much weaker. Vice versa. If you see a gene, it's very, very green, not red, it means that particular gene is highly expressed in normal cell, but not in cancer cell. Okay? If you see a yellow spot, that means equal contribution, right? You have red, you have a green, so kind of equally combined. So if you have half green, half red, then it displays yellow. Okay, so that means that particular gene, their expression level, there's probably no difference between cancer cell and normal cell. So based on their fluorescence signal intensity, we will be able to tell which particular genes are highly expressed in cancer cell, which one is very, very low expression in cancer cell, and then which are those genes, there's no difference. So based on up and down, so we will be able to study why these genes are highly expressed in cancer cell, or why these genes are highly suppressed in cancer cell. And then, of course, later on we have some bioinformatic tools can help you to understand why these genes are coming from. Okay, what's their function and which pathway they are belonging to. Okay, so using this, we will be able to screen many, many genes at a time. Remember, I told you we print 20,000 genes in 16 hours. So our in-house printing will have 20,000 genes. How many human genes do we have? Well, you know, we have slightly less than 30,000. Usually we say 20 to 25,000 genes, okay? So definitely not over 30,000 genes. So used to, when you want to study something, you probably have to study one gene at a time. But with this microarray technology, you will be able to study gene, you know, tens of thousands of genes simultaneously. Okay? So that's why once you get this image, you need to convert this fluorescence signal intensities need to be converted into a numerical data. So I will be able to know, okay, how red they are, how many folds increase, or how many folds decrease compared to normal cell. Okay? And then based on this, then you will do the interpretations. If you see some genes up, some genes down, what are those genes? What's their function? What's their biological meaning having some genes up and some genes down? Okay? So nowadays, it's very easy to, as long as you have money, you have funding to support, you can do whatever experiment you want to do. But how to interpret it becomes a daunting task. Right? If I give you 5,000, for example, you use 20,000 genes to screen, and then I find out what you find out. There's a 400 genes difference. 400 significant change gene, okay? Then you have to figure out why are these 400? Which one is more important than the other? Then we have to use bioinformatics to analyze and to interpret these data. Okay? So, am I clear? The whole process from the production to analysis. This is the whole semester we're going to focus on this. Of course, like I just mentioned, we're going to talk about NGS. So NGS is part of the sort of biochips in general. Okay, they're still using a biochip, microfluidic channel, but using slightly different technology. But again, these are all how we call high throughput genomic technology, because you can study many, many genes at a time. Okay, high throughput, in Chinese we call 高通量, right? Like I said, in the past, if you want to study 1 gene, 2 gene, 3 gene, maybe 5 gene, 10 gene, that's the most. But nowadays, you know, if you want to study many, many genes at a time, then this is one of the methods. And the other one is the next generation sequencing. Okay. So, microarray service. Remember I told you why we picked glass. There are three types of, in the beginning, people are using membrane. Okay? In Chinese they call it 滤膜. Second is glass. Third is the silicon. Glass 玻璃大家都知道了 I don't want to say that in Chinese. So, in the beginning, I think probably in my first lectures, or I really can't remember. So, microarray technology actually, in principle, it's based on a technique we call southern blot analysis. I'm sure anyone doesn't know southern blot analysis? You all know that, right? Oh. Southern blot. It's a way to study, to identify a known or the unknown probe. Okay? So, regardless whether southern blot analysis, northern blot analysis, or western, people are using membrane, loops, right? Either you can use nylon membrane or you can use a nitrocellulose membrane. So that's the membrane. So when people start making array, they're actually using membrane as their first subject. Okay? So, and why people start sort of giving up later on? Because of some of these problems. Okay? So they move to glass or silicon. Membrane is like a white paper, right? I'm sure you all have some experience. If you have some water spills on the paper, once it dries, you'll see something happen, right? You don't get the flat surface anymore. You will see some wrinkles, right? 皱褶，就纸张撕掉以后就会变皱褶。Okay. So if that's the case, then you won't get the flat surface. Okay. Planar 跟 flat 是差不多意思。好，就是你需要有一个平整的表面。Dimensional. It means, I told you, we have to print the DNA onto a specific area. And then you use this glass is 1 inch by 3 inch. Okay? And then the spot need to be very uniform. Okay? You cannot have a big spot and suddenly the adjacent spot become very small. Okay? And then some of the material you use need to be durable, because you see here, we have to go post-processing, we have to do coating, we have to go through all kinds of chemical reactions. So you have to make sure some of the material you use need to be durable, okay? And then inner, it doesn't itself, because remember, we use a fluorescent dye. A lot of things will have autofluorescence. So you don't want to have a higher background. Okay, you want to make sure the signal is really coming from the DNA itself. It's not coming from something else. For example, the fiber, or coming from your hair, or coming from something else, the dust. Okay? So, and then of course, you want to make sure the binding is very efficient, and then, it's accessible. This is how the textbook lists all the parameters. If you want to find something new, then you have to consider all this. Of course, over the past 20 years, pretty much everything is done. So nowadays, you just buy something directly. You don't worry about all these different factors. Okay? So dimensional, I already told you, 1 by 3 inch. This is the typical microscopic slice, right? And some company, they have a longer, like we may discuss one company called Illumina. The array is slightly longer, slightly narrow, okay? But typical size is 1 by 3 inches. Okay, so that's what I say, 2.5公分乘以 well, in this case, it's called 7.6公分. This is one of the, this is the largest microarray company, or biochip company in the world, called Affymetrix, okay? Their chips they are using silicon. They are not using glass. They are using silicon here. And then their dimension is 1/2 inch by 1/2 inch. It's kind of square. I told you our in-house printing is about 100 micron. This is even smaller. Okay, we will discuss that later on. Okay. So different company may produce a different chips with a different size. Deviation from flatness. Because if it's not flat, they will impair the quality of the array printing. Okay? Then it will also affect your detections. So in this case, right? If you have a flat surface, everybody is the same distance. The size may not change too much. But if you have an even surface, then you might have a different spot. See, this is the distance is longer, so you have a bigger spot compared to this one, and then this distance is smaller, so the spot is smaller. And then when you catch the signal, that will also affect the intensity. So not only the printing quality, also the detection quality as well. Okay? So that's why initially everybody using membrane, but after a few years, not anymore. Because I told you, membrane is like a white paper. Once you have water, you get wet, then you don't have the flat surface anymore. Okay, you're gonna see a wrinkle. So once you see a wrinkle, then you're gonna see something like this happen. Okay, so no one using membrane at all. Of course, some companies they use glass on the bottom and then put the membrane on top. Okay, because you need to get a good support for flatness. You see, yeah, during the detection, the flat surface, everybody getting the red signal. But in this case, if you have an even surface, then you have some red, you have some green, you have some yellow, because the signal varies by detection devices. Again, this is a very small spot, right? So you need those scanner I just showed you here. It's a very small device as well. So the lens, how you collect, do the excitation and emission, collect the signal, it matters. Okay? And then you don't see this is a small one. This is more like a small laser printer you have at home. But this scanner had 2 solid laser inside. We will discuss that later on in the chapter 7 or 8, I cannot remember. So there are 2 solid lasers inside, and then this particular scanner is very compact. It will cost about 1.8 million NT. 180万一台这样的 scanner. Okay, so you see why, and then of course, if something like this happens, it's supposed to maybe you have 400 genes on significant difference between cancer cell and normal cell, right? But if something like this happens, your result is totally different. And this kind of result is totally biologically meaningless. It's not coming from the biology itself, it's coming from the device problem or your array problem. Okay? So that's why we try to minimize. That's why it says flatness is so important. One of the factors you want to make sure this kind of deviation won't happen, at least in your array. Well, planar is, like I said, is something like a flatness, same thing, okay? So substrate level to within, so they allow some deviations, so plus minus 10 microns, okay? So to printing and detection, so this is okay. So plus minus 10 microns, okay? And of course, you want to make sure it's not tilt. Remember, membrane, no good, right? We don't use that anymore. Glass is okay, silicon is okay. You don't see plastic. Why we don't use plastic? Because plastics may see this kind of problem here, right? 各位如果有用过塑胶，就知道有时候塑胶加热它会跳，那你就不是一个平整的表面。Okay, so that's why plastic is not on the list. Glass is very stable, silicon is very slow. Even you heat it up, it doesn't hurt. Okay, but plastic, if you heat it up at high temperature, you might change the surface. Okay. So you want to make sure the x and y axis they don't, you don't bend or something like that. So you provide the inaccurate readings. So uniform. In this case, that means the reactive groups per unit areas. Remember, let's go back to this slide here. We have to do coding, right? So what kind of chemical you use? Are they good enough? How do we judge whether this particular chemical is good enough? It's based on per unit area, their reactive group. Okay, let's come here. So in this case, it's evenly distributed, A, B, C, D, but this is not uniform. So one particular area have a more reactive group, so you have more DNA printed on that particular area. Compared to some other area, you don't have any DNA printed. Okay, then that again, not gonna give you an accurate result. Okay, so you want to make sure the chemical you use for service, either treatment or coatings, it has to be very uniform across the entire slide. If you are talking about the whole slide, so that means the whole slide need to be, very uniform, right? You cannot have one slide like more than the rest of it. Okay, so that's how this figure implies. Okay, I just mentioned, what is the surface treatment or what is surface coating? What's the difference? Okay, treatment is chemically reactive monolayer formed by covalent linkage. The keyword is covalent linkage. Okay, of this particular reactive group onto the substrate molecule. The substrate molecule, in this case, we usually say glass. 意思就是你用的化学物质是直接可以跟玻璃产生化学反应。 OK， 而且是透过 covalent bonding、covalent linkage。好 ，and I think Professor Tsai is supposed to cover chapter 2. Chapter 2 will discuss about 4 different chemical bondings, right? Let me ask you, what's the strongest bonding? Covalent bonding, right? What's the second one? No. Ionic. 离子键. And number 3 is hydrogen bonding. And what's the weakest bonding? Yes. Yeah. Van der Waals. Okay. So covalent bonding. 所以它会跟玻璃的表面产生化学结合，就说你用的这个所谓的化学物质，它会跟玻璃表面。And how that different from surface coating? Coating means it's just a thin film placed on top of the microarray substrate in the non-covalent manner. 这就没有产生共价键的结构，就是你用的化学物质，它是在有点像这个，各位有没有车子？如果有车子，你要去手机你要去包膜，那是概念 ，right？ 你包的膜是不会跟手机上面产生化学反应，那就完蛋了，你撕都撕不掉 ，right？ So this is the surface coating and this is surface trim. Okay? So I told you the key word is covalent linkage. This is non-covalent manner. 好，这是表面。那当然 ，if you ask me which one, this one probably 比较容易就是被洗掉或者是被处理掉。好，所以 two types of either surface coating or surface trim. I told you earlier, nobody want to use the membrane alone, right? But some people, they're using membrane plus glass as a support. So you see those are Y square area. These are 8 blocks of the nitrocellulose coated soil. Okay? So you can still do it. Again, nowadays, probably nobody is using that. But in early days, some people start using, they kind of, because they don't want to use the membrane directly, so they put a glass. Okay. This is the way how we do coating in our own lab. Okay. We use a chemical called lysine. The full name is polylysine. So you see, having lysine and without lysine coated, you see 那个水是会聚起来。OK， 这个叫做 hydrophobic， 叫疏水性，所以它会把水柱拱起来，对不对？另外还有一种叫什么 hydrophilic， 亲水性，它就会散出去。Now, different chemical, they have a different sort of chemical properties. Okay, this one, lysine we use is hydrophobic characters. But you can use different chemical, they may have hydrophilic characters. Okay, so you see this with lysine, without lysine, water is running everywhere. You even can put a smile face here, but not in this non-coated slide. Okay. And for this particular chemical we use, there's a process called aging. Okay, you cannot, once you coated the slide, you cannot use it right away for printing. You have to wait at least 2 weeks, sometime up to 2 months. in order to build up this hydrophobic character. This is a kind of from our own experience. 就是用这种化合物啊 lysine 你就要让它放在这个平常室温下面要放2个礼拜，你才可以拿来用。Okay, is there a way to speed it up? Yes. 如果你要加快，就是要把它加温，放在一个比较高温的情况下面，让它比较快形成这个后面。OK。因为如果你没有这样子，它就不会聚在那里，到时候你打上去的点它就会散。Alright？ 好。Durable. It refers to the stability of the substrate and the surface treatment or coating over the duration of microarray experiments. Right? I told you in this slide. Not only we have to use this 3-dimensional robotic to print DNA onto the glass slide, we have to go through the post-processing process. We need to add different enzymes to do the labeling. Don't forget, messenger RNA need to go through reverse transcription. You have to use a specialized enzyme called reverse transcriptase. Right? So you not only have physical impact on the slide, you also have a different chemical impact from post-processing, from labeling, even from hybridization. And then hybridization, we will talk about that later. We also need to increase the temperature. So you also have some thermal effect as well. Okay? So that's why you have to make sure the chemical you use, the substrate you use, they can be very stable over this microarray experiments. Okay? So you can lose some of the molecules, but again, there's a benchmark, 10%. Right? Processing and reaction condition for microarray analysis. I told you, thermal effect, elevate temperature. We also need to denature the DNA. If you recall, in my first lectures, I mentioned that we may have 2 different types of DNA. One is single-stranded, the other one is double-stranded. Depends on how you prepare it, right? Single-stranded DNA, you print it on the glass slide, it's fine. But when you want to do the hybridization, you want to do the testing, you need to denature the DNA to allow single-strand DNA to bind with those labeled, the complementary DNA, when you label with fluorescent dye. So they can renature together during the hybridization, right? So, and then how to denature DNA? DNA, remember, the double helix structure is holding by hydrogen bonding, right? Hydrogen bonding in the 3rd weakest, well, I should say, it's a 3rd weak binding within the chemical reactions. So they can easily be breaking up by heating up, right? When you do the PCR, I'm sure pretty much everybody knows PCR reactions. So you heat it up first, so you denature the double helix structure, and then you start do the primer binds in, elongation, and then close up, and then heat it up again, right? Repeating cycle. Same thing like this here. And then I told you, we have a lot of chemicals. There's organic solvent, like alcohol for dehydration. During the hybridization, we have to add a special chemical called formaldehyde. Okay, So these are all organic solvents. So that's why I told you, you might have physical impact coming from the thermal effect or coming from the printing. You have this chemical or from enzyme you use. Okay? So these all will affect your reaction condition. So going through this high temperatures, organic solvents, you see, well, this is quite durable, right? Or you print the DNA still attached to the glass surface. No problem. 0 hours, 3 hours, 6 hours. And then start binding coming from this complementary DNA. But this, if you have an unstable surface, you start losing some of the printed DNA on the surface. Okay? Anyway, I mean, the substrate you use, it doesn't have the autofluorescence or doesn't react with the DNA itself. Okay? So the definition is the microarray surface binds target molecules in a stable manner, but does not contribute any gain or loss of signal. I told you, don't have any autofluorescence. If you use something, they will have fluorescent, then it will affect what's the signal intensity between red and green. Okay? So that's why it doesn't gain or loss of signal in the assay. Okay? So come on bench flow. The substrate and the surface treatment or coating that each contribute less than a twofold additional signal relative to the signal obtained from a detector without a loaded microarray. So that means, regardless what kind of surface treatment, for example, if you use this lysing on the surface, it doesn't contribute too much compared without lysing. Is that clear? Right? So the chemical you use cannot, contribute to the signal. So that means it will have a higher background. So when we were studying how good is, we will discuss that later, but you have to remember one acronym called SNR. Okay? If you are coming from the communication background, then you know this. So this is called signal over noise ratio. Right? 中文就是杂讯，就是你的讯号跟你的杂讯之间的 signal over noise ratio. If it's too low, that means your background is very high. If your background is low, then your signal is good. That means SNR will go up. Okay? Yeah, so the chemical, whatever you use, you don't want to then contribute to your noise. Okay, make sure that's not going to be the case. So either elevated or low readings, okay, so different, like, see, I already told you, background fluorescence caused by the chemical used to prepare the surface treatment or coating. So that's a no-no, right? You don't want to use something. It will increase your background fluorescence. And of course, light scattering, I already told you, dust or other contaminants, okay, maybe during the microarray manufacture or use. So that's why you see that's three-dimensional robotic, actually it's in an enclosure. It's not in an open space. Because in the open space, you have a dust. You might have some fiber coming from your clothes. You might have even some hair going during the production. So you need to have a, and then we will discuss that later, saying you need to have a very good climate control. Temperature, humidity. That's another two key factors. Okay. I will discuss that with you later. Okay? So, low readings, again, I told you we use a lot of enzyme, right? We use some organic solvent, chemical agent. That will reduce the microarray signal, either by damaging the target or pro-molecules during the microarray analysis. That means it will damage either you already printed on the surface, or you will damage those trying to be, linked to the printed DNA on the glass. So you want this to happen, okay? Efficient, okay? So you want to make sure the efficient with this surface treatment or coating can bind to the printed target, right? Once, if you use some chemical and then you print it, it cannot print on the surface, then this chemical is not good, right? So you want to make sure HDNA, for example, if you print maybe 100 copies and then at least maybe 90 copies are already onto the slide, okay? So, and of course, the target binding efficiency is in the range of 10 to 30% for a target mixture, okay? So you have to find a minimum, optimal concentration. Too much is no good. Too little is no good too. Okay, we will discuss that. And then say binding efficiency need to be measured and adjusted empirically. Yeah, this is, one of the in Taiyu, so this is one of the main gum. It's not just you take the DNA and you print it. No. You have to make sure it's the right concentration and then how you print it. It's a lot of makeup actually. So accessible, that means you make sure it provides a productive interaction for the target with the interaction between target and probe, which means here, right? You provide enough, they can bind. This is the arrow, down arrow. Okay, this is what you printed on the array. This is need to be tested here. You want to make sure this is reduce the accessibility can be compensated. Again, this is how you judge, how you prepare your slide. So I think it's quite straightforward. So optimal target concentration. Like I said, you don't want to go too high. You don't want to go too low. You need to find a right concentration. If you go too high, see, this is a fluorescent signal. You will drop. And then too low. And then of course you have to make sure it's in the linear range. Okay. Sometimes you go over to get hot cold. So this is the figure from our textbook showing that 30 micromolar is the optimal. Of course, you still, again, you have to adjust empirically. So you have to fit in your own conditions. May not say, oh, that's the standard. Those standards, no, that may not be the case. And also how you prepare your DNA, that's another issue. Okay, why we put this here? DNA structure, they have a major and minor groups, right? Because this is hydrogen holding double helix structures. Okay? So you have major groups, you have minor groups. So you want to make sure this, when you do the array, the hybridization, they can perfectly match to each other. Okay? Well, this is, quickly go over this. It's because of this. The DNA double helix structure is about 20 angstrom, the width, right？ 就是 DNA 这个双股螺旋的结构，它的宽度大概是20个angstrom。So when you print the array， 你不能太密，你太密，你给它足够的空间，让它去做这个 hybridization。Remember, we print one DNA onto the glass slide, right? So we have another DNA comes in， 然后利用我们常讲的叫做共轭互补 （base pairing） through hydrogen bond, right? So this is the target, this is the probe. Okay. Well, I have to explain to you in our textbook, they kind of using this, the definition for target and probe is somewhat slightly different than our common practice. Okay? Target in this case means the DNA, the known DNA. You print it on the array. And the probe means those messenger RNA be converted into a complementary DNA, which is a known. Right? But our common sort of concept is probe should be the known DNA. If you adopt it, remember I told you the southern blot analysis, you have a known, you run the DNA through the geoelectrophoresis, you print it on the either nitrocellulose membrane or nylon membrane, and then you take a known probe, a DNA sequence you already know, to hybridize with the membrane and try to find out on that particular membrane which one is the one you are interested in. Okay? So to kind of elaborate a little bit, so for those DNA fragment we already printed on the array, it's a known DNA fragment, right? It should be called probe in our sort of perceptions. But in our textbook, kind of using the opposite, say they want to put target, and then probe is unknown. Okay? So, but if you see only for this textbook, so you know, don't get confused. You might see some papers showing that, oh, how many probes we already prepared. We print, for example, 5,000 genes. We have to prepare 5,000 probes on the array. Okay, so the common practice is those printed on the array should be called as a probe, and then those need to be tested, or the assay should be called target. But you know, I don't know, Max Shanna, he just insists, he feels this is much better, so we just use target for those. Anyway, so you need to provide enough spacing to allow this base pairing to happen. Okay, so in this case, 20 angstrom. Now you only allow 10.8, even smaller than 20, right? So you don't have enough spacing for this double strand binding to be happening. So that means you can only bind to the one with enough spacing. For those 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, you cannot bind to it. Okay? In this case, that means your signal gonna be underestimate. It's supposed to bind to 12, right? But only binds to 1. So the signal gonna be very, very weak, because you don't provide enough space for them to form this hydrogen bonding. Okay? So that's why if you print it, the concentration is too high. See, the signal is actually dropping because too much DNA. And of course, if you too far away, again, the signal is going to be weak because you don't have enough, whatever you call a target here, right? 20, at least you can put another one here. You can put another one here. So that because the concentration you prepared is too low, then that's the situation you will be facing. Okay? All right. So, this is the probe concentration, this is the microarray signal, right? Need to be in the linear range, and then you have to find out this optimal probe concentration. So you won't want to add too much probe as well. In this case, it's your complementary DNA. You don't want to put too much as well. Okay? Any question so far about this one? Okay. Okay. So you want to make sure actually you have enough DNA on the glass line. Of course, make sure it's not going too high, otherwise you have to allow space for binding. So that's why you have it in a linear range of 1x, 2x, and 4x, right? If you're adding too much cDNA, then you're going to see something like this. This is already saturated. They occupy all the binding sites. Okay? So that's why you don't want to use too much of the probe as well. So sometime, I think maybe later on we will have some picture showing you'll see some white spots. That means it's already saturated. Okay? Because this is fluorescent, it's kind of, it's, we call, it's a 16 bits signal, okay, in terms of either green or red fluorescent. So it's that's the maximum signal intensity, 65,536。2的16次方，因为它是2位，它是16 bit， 所以荧光的强度最强就到6536。You cannot go anything above. Okay. So once you go this, they still remain 6536. It's already saturated. 已经饱和了。So, what kind of situation you will be facing is, here, for example, okay? Then if you add too much, then this is supposed to 5-fold difference, right? It's already saturated. This one is not, so it's increased 3 times, but this is only like slightly 1 point less than 2 times. So in this case, it's only down to maybe 2.24. So if you add too much, then actually it does not reflect the real biological changes. It's supposed to change 5-fold. 就说这个基因的表现，原来在肿瘤组织在癌细胞里面跟肿瘤细胞差5倍，结果因为你给人家加太多，结果最后只差2倍半，2倍多一点点。如果再差少一点的话，就被 if you filter it out, then you miss this particular. Okay, so that's why it's kind of very tricky. How you, every single step during the array, the cycle I just showed you, it's a lot of makeup as well. Okay. Yeah, of course, if you want to study the low abundance mRNA, then of course you have to add more of this. Okay, so low abundant, then you have to increase your probe access. 就是说，如果你要研究那种非常稀少的 messenger RNA 的时候，你当然要把量，不然太少的时候，它 signal 没办法转变出来。Too weak, maybe the signal is almost equal to the background noise. So you won't be able to tell the difference. So that's why you have to add a lot more. But in the normal situation, that's not the case. It will happen like something like this. Okay? So a different approach to answer different questions. If you want to study low abundance, then you need to have a probe axis. If just a regular abundant messenger RNA, then you have to make sure it's using this. Yeah, so this is a 1x, 2x, 4x. The stronger, right? The more concentration, the higher the signal. And then selective circulation, so it's not quantitative. So the good thing about the microarray is not only it's qualitatively you see some signal, it also give you quantitative measurements. How much fold increase or how much fold decrease, right? They show you here. Okay.